Hi everyone, and welcome to the third TIA Centre seminar of the academic year. So my name is Adam Shepherd, and I'm a researcher here at the TIA Centre. And for those that are new to our seminars, we invite researchers and leaders in the area of computational pathology and associated areas to discuss their work. I've also now got a co-host, Jachi, who's now helping running the seminars and organising. So I don't know if you want to just put your camera on and wave. Fantastic.
Yes, yeah, hi everyone. I'm Jachi. I'm helping Adam to run seminars. Started it recently and hopefully to take on more tasks in the future. Thanks.
Brilliant, thanks, Jachi. So, but today I am delighted to introduce Malte Kuehl. So, Malte studied medicine at the University of Hamburg, where he joined the Institute of Medical Systems Biology and the medical department to begin his Doctor of Medicine degree. In 2023, he joined the Complex Tissue Lab at Aarhus University in Denmark, led by Professor Kuehl as a postdoc. His primary research interests include spatiomics, image analysis, and agentic AI. And the title of the talk today is Scaling Multiplexed Protein Imaging Analysis for Subcellular-Resolution Pathology. So Malte, thank you again for joining us, and we're very much looking forward to your talk. And with that, I'll pass over to you.
Yeah, many thanks, Adam, and the whole team for putting this together and the invitation. I was really happy to receive it, and yeah, I hope that you can also get something out of this presentation, which I'll share in a second. Okay, can you see my slides?
Yeah, we can see it. Okay.
Perfect. Yeah, and yeah, as you mentioned, the title of the talk is Scaling Multiplex Protein Imaging for Subcellular Resolution Pathology. And I won't go through my affiliations again, since you already mentioned them. Just perhaps for kind of how we do this meeting. If at any point you have questions or want to ask something, feel free to interrupt me. I didn't know exactly what the background of everybody here was, so I assumed, for example, that everybody knows how immunofluorescence worked and didn't go too much into the details of some of the technical things. So depending on if you want me to dive deeper into something or want me to explain a concept that might be a bit out of your field, just feel free to interrupt anytime and we can adapt it. So with the title of the talk, I think the focus of our lab is mostly on complex tissue, as the title of our lab suggests. And if dealing with complex tissues where you might have interactions of many different cell types, many different pathological processes, and also regulation at different levels, I think the motivation for spatial technologies is clear. Because while with bulk proteomics or bulk transcriptomics, you can identify perhaps global processes or processes that are just within one cell population. If you have in vitro data, and similarly with single cell data, you can get cell level population processes. If you really want to understand what is happening in pathology specimens, so in complex tissue derived from human or animal models of disease, you really would want to go to spatial technologies to really understand what cell types are interacting with each other, how they are arranged, and what is happening at the organ level. However, what might be a bit less clear is why in our lab we focus mostly on spatial proteomics rather than spatial transcriptomics. Because if you go to PubMed and just search for spatial proteomics and spatial transcriptomics, you can see that there's about an order of magnitude in between the references. And of course, spatial transcriptomics has shaped the field of spatial technologies and our understanding of tissue in the recent years and has uncovered many new insights into tissues. However, we also think that spatial proteomics and particularly multiplex protein imaging can provide further and additional insights that can complement this. And the motivation basically comes from the central dogma of molecular biology, because while all your genetic information is encoded in DNA, there are different levels of regulation. Some are epigenetic and at other processes that regulate the transcription, which leads you to RNA, but also between having your RNA and having your proteins, there are different levels of regulations through half-life, through further through post-translational modifications and just through kind of how many RNA molecules you need to arrive at a certain number of proteins. So while RNA can already give you a lot of information, if you want to understand the phenotype of a cell within a tissue, a protein really has a lot of additional information. And in this context, RNA can perhaps be seen as the potential of what can be manufactured and kind of the direction, because it happens on a shorter time frame where you have more bursts in transcription, and the half-life of RNA is typically much shorter than that of proteins, while proteins and specifically post-translational modifications allow you to understand the current composition of a tissue and its molecular function. And this complementary addition through proteomics and the potential to gain new insights has of course been recognized, which is why spatial proteomics was named the method of the year last December by Nature Methods. And yeah, talking about spatial proteomics and particularly multiplex protein imaging, which is kind of the subfield of spatial proteomics that we mostly focus on, perhaps it's best to understand it by understanding what came before it. Because if you want to examine the spatial expression of proteins, both in a research as well as in a clinical setting, what is often mostly used is either standard immunofluorescence or immunohistochemistry. But due to limitations of these technologies, for example, with immunofluorescence, spectral overlap and cross-reactivity, and yeah, just availability of different antibodies, this is typically limited to about 2 to 5 markers or 2 to 5 proteins or structures that you can have within the same sample. And this limitation in recent years has motivated the expansion of what is possible and the expansion of marker counts to basically form the field of multiplex protein imaging, which refers to any field, to any technology that can scale kind of beyond these 5 markers. And yeah, this has been around now for already a couple of years, and there have been different technologies. And a good overview of existing technologies was provided by a recent publication last year from D'Souza, from the Bodenmiller lab in Switzerland, because there are different technologies that you can use. The first group of technologies kind of is the mass spectrometry-based one. And here you bind your antibodies that target specific antigens within your tissue to rare metals, and then stain your tissue in a single step. And then you do not image them optically, but instead you use a UV ablation or ion beam to typically ionize parts of about 1 square micron sized areas in your tissue, and you link this to time of flight readouts, so kind of CyTOF, to understand where these metals that you know correspond to specific antibodies, since these metals do not naturally occur in your tissues, are positioned within your tissue, and then you can reconstruct an image of protein expression that way. And this has already been commercialized and has been around now for, I think, more than 10 years. And has been shown to, yeah, widely be replicable and useful. Another group are the microscopy-based technologies that actually use optical imaging. So you can use them with either like your own microscopes, or if you have a commercial system, because some of these technologies have also been commercialized with the microscopes and optics built into the commercial systems. And here you can kind of differentiate 2 subgroups. One is the fluorescent tagged antibody group, where you either directly have your primary or your secondary antibodies that are bound to fluorescent molecules, and then you perform basically standard immunofluorescence. But you can do this in cycles by either deactivating the fluorescent molecules that are in your tissue or directly removing the antibodies from the tissue to then get unstained tissue again and be able to do this in iterative cycles. And then a variation of these are the oligonucleotide-based methods, where instead of staining the entire tissue with like 1, 2, 3 markers at a time, you stain all the markers you have at the same time, but instead you change a complementary sequence called an imager for the different oligonucleotides that are bound to the antibodies to then get an iterative readout over multiple cycles that way. But the basic concept is kind of the same, that you only acquire 1 to 3 signals at a time and iteratively go through this. And if you look at the scale that has been achieved with this, typically in terms of pixel resolution, this lies within the range of about 200 nanometers to 1,000 nanometers per pixel. And in terms of marker counts, most technologies are in the range of about 30 to 60 different proteins or structures that you can image within the same sample. And yeah, if you want to see how this looks like, here's an image from imaging mass cytometry. So one of the most established techniques here with a 1 micron per 1,000 nanometer pixel resolution. And you can see that this gives you very specific and good signal, and where you can clearly distinguish that there are nuclei that you can segment. And then this has also motivated the kind of standard analysis that is typically performed on these tissues, which is cell-based, where you identify where your nuclei are located, or if you have some membrane staining, even perhaps the cytoplasm, and then you perform your analysis at this cell level. And with this cell level analysis, there are different possible analysis you can do. You can identify the cell identity, so where you have different populations that you might have predefined and might have used specific antibodies for in your panel. You can identify cell states, so for example, which states are MP67 positive and might be replicating, or which cells might be injured. And since you have these spatial contexts, you can also profile cell-cell interactions, or at least co-location. However, this has been very broadly useful, and there are many publications on different organs with the different technologies where this has been used to uncover novel biology. But there are some layers of biology that are not fully covered by it, because those layers require a subcellular resolution. And part of this would be identifying cell organelles and where these organelles are located. Or if you think about the extracellular matrix, protein co-expression within the extracellular matrix. Because of course, if you segment the entire extracellular matrix, it might be very diverse, and you might not be able to really identify co-expression patterns because the mean might not be very meaningful over such a large area. And when it comes to signaling within a cell, it might also be important, for example, if you have a phosphorylated target, where it is located within the cell. So for example, if it's in the cytoplasm or in the nucleus, can have different implications on how you would interpret this biologically. So to kind of add these layers on top of what already exists, you need subcellular resolution, and also to perform your analysis at a subcellular scale. And this has basically served as motivation for us to identify a range where we thought we could add some value, where we could use confocal resolution, which typically has not been used with these kinds of technologies, and also scale in terms of the marker counts to cover more processes, and to the cell identity and cell state layers, add more things that also cover subcellular compartments and more cell signaling. And yeah, here's an example how confocal resolution looks like, of a very similar structure as I showed before. And you can see that you can clearly distinguish the membrane, the cytoplasm, and even inside the nucleus, different nucleoli, and that you have resolution that allows you to do analysis at a different level. And to do this in a multiplex protein imaging setting, we came up with PathoPlex, which is short for pathology-oriented multiplexing. And we were lucky enough to have it published in Nature earlier this year. So while today I will present some of the imaging optimizations and some of the technical analysis and bioinformatics that we did, I cannot cover everything that we presented there, and I will not go deep into the biological results that we obtained on different data sets. So if you're interested in that, then please feel free to check out our publication as well. Yeah, and with PathoPlex, we conceptualized a concept called integrative mapping, where we try to assess all these different layers of biology that I presented previously, going from the multicellular architecture down to the subcellular organization, and covering cell identity, but also subcellular location, and importantly also signal activity with, for example, also some phosphorylated targets that might not even be detectable with some of the other methodologies, because sometimes you require amplification to see this. And our PathoPlex framework builds on two pillars. The first is that we wanted to really scale subcellular resolution multiplex protein imaging, both in terms of the resolution, but also in terms of the marker count. And then derived from this first scaling, we also needed to scale the computational analysis because we would acquire a lot more pixels and a lot more markers. And we also wanted to analyze things at a subcellular level and not average them at the cell level to get out some of the information that we actually imaged without losing it. And there's some pre-existing work in this direction. And in some sense, PathoPlex represents an evolution, extension, and reconceptualization of 4i, which was introduced in vitro and on a smaller scale by Gabriele Gut and others from the Peltman's lab in 2018. And they've been collaborators also on this project, which now has focused on large-scale tissue studies with pathology specimens. And on the imaging side, our protocol is a framework based on illusion-based multiplex protein imaging. So you start with unstained tissue, then perform basically standard indirect immunofluorescence, and then you can acquire your signals of up to 3-4 different proteins at a time. But then, since these antibodies don't actually bind covalently to your tissue, at least if you make sure to also use some free radical scavengers to prevent cross-linking, you can actually elute these antibodies through pH modulation and through washing buffers to go back to unstained tissue, which allows you to repeat this process many times. And here we were also really focused on democratizing access to kind of this next generation of multiplex protein imaging, because all the optimizations that I will present in a moment that we did were focused on working with archival formalin-fixed paraffin-embedded samples, which are the most abundant and most easily accessible type out there, because while some technologies work better with fresh frozen tissue, you really need to plan this and cannot go back to a biobank that might exist, and these samples are generally less available than FFPE samples. It also works with commercially available standard chemicals, and you can bring your own microscope. So while it works with high-resolution confocal imaging, we also have done some studies using wide field or spinning disk microscopes. So even if you have just the wide field microscope available within your lab, you can actually use it with this technology. And importantly, you can probably also reuse all the antibodies that your lab might already have, because we use off-the-shelf antibodies. So if you ever perform standard immunofluorescence or standard immunohistochemistry, it is likely that unless you require a very specific pH or super long imaging times or something super rare, that you can actually reuse them and that they will work with this protocol. And additionally, even though you have this flexibility, still allow you to be high throughput, we came up with some optional 3D printing based high throughput add-ons that I will also present later on. And with this imaging protocol, we really were able to achieve a new scale where we were able to stain in many different samples on the same plate, more than 120 proteins and more than 20 additional quality controls over more than 90 cycles within each sample, so within each specimen, at 80 nanometer a pixel, for a total of more than 500 billion pixel intensities in the largest experiment that we did as part of this manuscript. And yeah, this really allowed us to acquire a lot more information at the same time, but also posed some challenges, because now we had this new resolution, we had this new scale in terms of markers, but we also needed to analyze it. And to get out some information at the subcellular level, we conceptualized pixel level clustering, implemented through a Python library called spatiomic that I will present later on. Basically, the concept here is that, of course, you acquire your images iteratively with about 2 to 3 markers each cycle until you reach, in this case, the 120 markers. But of course, you can also just do 30 or just do 60, depending on what you would like to profile and how, the variation of the tissue that you're looking at. And then you perform first some image registration where you match and align the images from the different cycles so that you can interpret them together. Then you just pre-process the signals to enhance the contrast and remove perhaps some outliers. And then the main part here is that we perform pixel level cluster generation, where through self-organizing maps and KNN graph and Leiden graph clustering, we are able to cluster and assign identities at a single pixel level. And this might sound a bit abstract, but if you see an image, it will hopefully become clearer. Because here, this image is fully segmentation free. This is just based on pixel level clustering, where we just use as features the protein co-expression at every single pixel. And here you're looking at a kidney sample where you have a glomerulus in the center where you can tell apart different cell types. And then here there are some proximal tubular structures where you can see that in the cytoplasm, there are different metabolic markers that we had here. And then at the more apical level, you can see the brush border forming, while the nucleus and around the nucleus, you have different expression and also different parts of the tubule, like here the proximal tubule, or here parts of the distal tubule are colored in different ways. And this works without requiring any segmentation at the single pixel level with this kind of concept. And this really has some specificity to biological processes and really works at a subcellular level, as you can see here in some of the zoom-ins from kind of the largest data set that we did today. That was also part of our publication, where you can see, for example, that you can identify even some nuclear signaling or some very basal and specifically located processes within different cells that are captured by some of the pixel level clusters. But of course, you do not just want to do unsupervised pixel clustering without understanding what actually is changing in your tissues between health and disease, and without understanding what these pixel clusters actually mean. Because since it's unsupervised clustering, you need to assign an identity to them yourself. So we also came up with a framework to do this, where the first, sorry, was there a question?
I don't think there was a question.
Has somebody unmuted themselves, but
Yeah. No, I don't think so, but yeah,
okay.
please continue.
Yeah,
Actually, I'm so sorry. My name's Sarah. While you just had that sort of break there, I did just wonder if I could answer a question, because I'm so sorry I have to leave in 5 minutes, and this is fascinating. I'm an ex-histologist. I worked in histology for over 20 years, and it was before that this came to the forefront, and it's just fascinating. But one question, if I wouldn't, if you don't mind answering, just before I leave, is I wondered if, number one, what kind of artifacts you would get, if you get any, and whether it affects the tissue integrity after these sort of different steps that you're doing. So yeah, if you wouldn't mind answering that, I'd be very grateful.
Yeah, thanks for the question. I think a lot of this is actually covered by some of the slides that are still coming up. But just to perhaps give a short answer, we actually see that the tissue integrity is maintained, and that even after more than 50 cycles, you can restain the same structure and it's still preserved. And you can also do histopathology afterwards, and you can see that structurally it's well preserved. There are some artifacts that are just more inherent to immunofluorescent imaging in general than this specific multiplexing protocol that we have to deal with. And we have come up with some optimization techniques that I'll show later. But generally, this has been shown to be quite stable. And yeah, in the end, when we did the 90+ cycles, the reason we stopped was that we ran out of antibodies in our lab, not that we could not process the tissue any longer. Yeah, so I hope that answers your question. I think later
Absolutely.
there will
Yeah. Thank you. That's
information.
amazing. Thank you very much indeed.
Thank you. Yeah. So Going back to how you can then interpret these kind of signals, we came up with a definition based on contribution specificity and contribution strength of each marker to each pixel level cluster that allows you to identify kind of the top markers that have the highest intensity and highest specific intensity within each pixel level cluster. And you can combine this with a spatial projection of actually where these clusters are located to then arrive at a conclusion what biological process they likely capture. And to compare this between different conditions, for example, health and disease, you can rely on standard statistical testing and log twofold changes to produce a volcano plot where you can identify how the composition of these clusters changes even at the data set level over many different samples, and what clusters may be associated with a specific disease. And here on the right-hand side, you can see an example where, for example, looking at this cluster 19, which is colored in red, you can see that this is quite abundant in this diabetic kidney disease sample, but not at all in the control samples from the kidney. And at the dataset level in the volcano plot, it also has a very high log twofold change and is statistically significant. And then if you analyze what markers are driving this, you can see that this is AIFM1 and TRPC6, which together with the spatial location, which corresponds to the proximal tubule here. And no, you're not necessarily all kidney experts, but I don't think like the details of this are super important. We could identify that this was likely related to calcium signaling because TRPC6 is the calcium channel in the proximal tubule. And this is one example of a finding that you could get out with this. There are more in the manuscript that we published. But to get to this level of analysis and be able to scale to this many markers, we did have to introduce some optimizations. And I think here I'll come back to some of the points that Sarah was perhaps concerned about, and rightfully so. And the first of all is that we had to solve registration, because if you have 90 plus cycles, there's always a small shift between, and then even if you use kind of the built-in software of your microscope, it never matches perfectly at the single pixel level. So to do this, we actually evaluated different stainings that we could use as a reference and image each cycle. So 2 nuclear staining and 1 pan-protein staining called NHS-SSTAR. Our evaluation showed that actually you can use all of them to align your images. But overall, our recommendation that we came up with was to use NHS-SSTAR because it's a pan-protein staining. So it stains all the protein containing area in your entire sample. Only acquires a single application at the start of the experiment, and it's relatively cheap with good temporal stability. And importantly, if you deal with organs that are not fully solid, so for example, in the kidney, we have tubules that have a lot of empty space, it actually allows you to perform a foreground segmentation, because since it stains all protein containing area, this area that is NHS the negative has to be empty background. And that way you can already remove some artifacts that lie out of your tissue, and also lessen the computational burden, because you can just focus on the area that does contain tissue. And then another problem we have to deal with that arose due to the length of our experiments with partly over 90 cycles and repeated processing was tissue lifting. And tissue lifting really refers to the tissue detaching from the imaging slide. So here you can see, I think this is an intestinal sample before lifting, and then a couple of cycles later in the same spot, you got this image. So you can imagine that then you cannot really match any signals or cannot interpret or analyze this at all. So we tried out various things to get this under control. And in the end, we discovered that actually the Aptose coating agent, which is commercially available, can prevent most of this. So in the study that we did of more than 90 cycles using the Aptose coating agent to attach your tissue to the slide, prevented this kind of macro lifting and decreased it from about 15 to just 1%, which we think is within an acceptable range if you do such a large scale study. And additionally, you can actually use the NHS staining that I introduced previously to salvage regions where you might not have this drastic lifting like shown here, but you might have a couple of pixels that are not present either because you have a slight Z layer mismatch, or because there's micro lifting where only a small part of the tissue deattaches and the rest stays attached. But if you have a pan-protein staining in every cycle, you can actually, okay, are there any areas that in at least one cycle are not stained by this? And then you know that there must have been some mismatch or some lifting, and you can exclude those areas while still using the sample for your analysis. And then finally, the third kind of imaging artifact that we had to deal with, which I think many people who have worked with fluorescence know, are red blood cells that have an autofluorescence that can be quite high, exactly in the range where a lot of your antibodies and fluorescent molecules emit their wavelengths. So this can lead you to not be able to analyze your samples at the pixel level if you also have positive signal within kind of the red blood cell area. But we actually use the glycophorin A staining, which is specific to red blood cells, together with some threshold-based segmentation to segment these red blood cells out, and then you can remove kind of this layer of noise from your analysis. And yeah, with those optimizations, we could eliminate some of the artifacts that we had in our imaging. But another optimization that we worked on was actually to increase the throughput so that you can process as many samples and with as little hands-on time as possible, even though this is a flexible protocol that can work with off-the-shelf antibodies and with your own microscopes. And typically, when you're dealing with such studies, you would use commercial imaging chambers, which typically have between 2 and 24 wells, or even more if you go to very small TMA cores. But if you deal with like tissue specimens from pathology, typically it would be up to 24 wells. But this introduces some problems. First of all, when you have repeated liquid handling, you need to pipette into every single well, which, if you do it manually, can be quite error prone. And additionally, you might not be able to fit all of the specimens that you have in there, and you might have to cut out some area if you have larger tissue samples. So our solution to this, to make this less error prone, easier to handle with single well pipetting, and more flexible, was to introduce a 3D printed frame for an imaging chamber that you can combine with commercially available glass slides to create basically your own imaging chamber where you can have up to 80 biopsies on a single slide, and you can only have to pipette on top of the slide and can stain all 80 samples at the same time. But we didn't stop there, because since we already had a 3D printer in our lab and we're using it to print these imaging chambers, we thought, okay, and what if you can actually use this 3D printer to do the liquid handling for us? So this was partly inspired by a paper called HistoEnder, but we reconceptualized this for multiplex protein imaging, where we built our own solution containers, discard containers, and tissue chambers that we 3D printed with the same 3D printer that we then used for liquid handling. And while this image might look a bit complicated, I think the best way to show this is a video. So you can see here, and this is a speed up, but you can use the nozzle of the 3D printer to handle this, to remove liquids from your imaging chamber, and then you can have all your different buffers, antibodies, whatever, in the different containers, and it can automatically add the solutions, help spread them uniformly over the tissue, and then also later remove them again. So this can run fully in the background. And for example, we have a former postdoc that now is a PI and has his own experiments, but he's partly also a clinician. And the way he does it is, in the morning he comes in, he starts his 3D printer, then he works the whole day in the clinic, and at the end of the day, he picks up his processed samples and images them with just a couple of minutes or an hour of imaging time, and he doesn't have to take care of the liquid handling and can combine his work in the clinic with his work in research. and thereby saves more than 70% of hands-on time. So these are kind of the technological innovations that we had on the imaging side, but to also make sure that if we scale to the scale that we aimed for, we don't introduce any errors and we have good imaging quality. We also added new layers of quality control. The first of which is that we always confirm that we get full illusion every couple of cycles. And you can see this here at a whole slide level that, you can stain with specific markers and then fully elute them and you don't have any remnant signals there. And you can also see this here over more than 69 cycles, that even after 69 cycles with probably at that point more than 90 or 100 antibodies, you did not have any remnant signal from any wavelength, because this also was together with secondary antibodies. So if there had been any primary antibody bound to the tissue, you would have seen it here. So this gave us the confidence that we're actually just imaging what we want to image each cycle and that we're fully eluding the antibodies. And then related to that, we also wanted to assess, are the markers that we're using specific? Because if you work with immunofluorescence, you sometimes have some that don't really work or don't really give you any signal. And we also wanted to control the level of autofluorescence. And we came up with a way of just measuring this based on the intensity percentiles, where if you look at a range that would typically be within kind of the specific signal of a marker, here the 99.95th percentile, you can see that for the secondary only, so these non-staining cycles that we did, this was quite stable and just represented some autofluorescence in terms of intensity, which did also not increase with imaging time, but remained very stable. And then for the primary stainings that we use, of course, the specific intensities depend on which antibodies you use, the imaging time, how abundant your antigens is, but you can see that basically all of them were above this level of autofluorescence, with the exception of a couple that we then later evaluated manually by looking at the images, and there the only reason that these were like some rare phosphorylated targets that were present in less than 0.05% of the images. But this is a good level of quality control to also help you find proteins that you perhaps have to evaluate further or to uncover what may not have worked well. We also were able to show that we conserved the tissue structure and the epitopes very well. So for example, we stained for two proteins, eccocorin 2 and decorin, in cycle 9 of an imaging run, and then later again in cycle 70. So between those two cycles, we had more than 50 other cycles, probably 70, 80 different other antibodies that were bound to the tissue, a lot of liquid handling and processing. But since we fully removed the primary antibodies from the first staining and then later added other ones, after more than 50 cycles, you can see that the tissue structure was well preserved and that you essentially get the same signal in those cycles. And of course, still, if you discover something through your PathoPlex run, if you have some primary results, you would want to validate them further. And in our manuscript, we added different layers through IMC, through single-cell RNA sequencing, bulk RNA histopathology, which can actually be performed even after doing PathoPlex on the same tissue, and of course, other validations through multimodal experiments. But with this, I would like to wrap up the imaging side of PathoPlex and focus a bit on the computational analysis, and mostly our package spatiomic, which we developed for PathoPlex, but also have now been working on to expand beyond PathoPlex and allow you to just perform different multiplex protein imaging analysis with it. And spatiomic is the Python package that is built on the scientific Python stack. So you can use familiar data structures that you would use with other packages. It's optimized for large-scale and multi-level analysis, so not only the subcellular level, but also combining different layers of biology. And it is optionally GPU accelerated. So if you have an NVIDIA graphics card, this can actually scale to many hundreds of billions or even trillions of pixels in just a few hours because it can use GPU acceleration. We have some documentation and extensive hands-on tutorials that are located at spatiomic.org. So if perhaps you think this might be interesting for some of your work, please feel free to check it out. And if there's any feature that's missing, also please feel free to reach out anytime or to create an issue on GitHub, and I'll be happy to implement it. But just to give you an example of what is possible with spatiomic, this is the basic code that is needed to perform pixel level clustering. So you can see we have some functions that allow you to load in images from different formats. You can then perform different types of pre-processing of the different channels, histogram clipping, Z-scoring, range normalization, logarithmic transform, etc., etc. And then to identify your pixel level clusters, you can train a self-organizing map through machine learning to basically get a representative example of signals that are contained within your tissue. And then you can use graph-based Leiden clustering to identify in an unsupervised manner the clusters that are contained within your tissue. And then we have some visualization of the quantification utility functions contained within it. So with just those 10, 15 lines of code, you can go from a raw image to this. So here, this is just an example where this code integrated more than 27 layers of different proteins, again in a kidney sample, and you can see here that you can clearly distinguish the glomerulus. Then on the outline of the Bowman's capsule, you have PECs, so parietal epithelial cells, but also this is not just at a cellular level, but for example, you can see here this thick membrane that is actually a thickened glomerular basement membrane that likely has a lot of collagen. So this is not, this is extracellular signal. And if you look at the proximal tubule, you can see kind of that there seem to be 3 layers corresponding to kind of the apical, center, and basal parts, which you would also expect to have different protein expression. And while this is optimized for PathoPlex, this is actually technology agnostic. So here are some examples that you can even use this with immunohistochemistry and codex data, and it still works, and you still get kind of this molecular specificity at the pixel level. Of course, what you can get out of it will depend on your marker panel, and with a 1 micron resolution, you will not be able to do the same subcellular level analysis. But for example, if you're interested in profiling the extracellular matrix in some IMC data that you have that cannot be covered by cell level segmentation, this is also something you can use to identify different clusters contained within that. But we do recognize that the cell level has a lot of information as well, and is a valuable level of analysis. So spatiomic also contains codes and utility functions to do cell level analysis, where we have a binding to the cell-post-SAM model that you can use to go from your tissue, typically your nuclear staining, to your cell masks. And then we have some utility functions that allow you to extend your masks and to perform cell-level clustering. And importantly, the cell-level clustering can be either based on the expression of raw or transformed immunofluorescence markers, but we also have some utilities that allow you to actually, for each cell mask, quantify what pixel level clusters are contained within those cells. And then you can do a clustering based on those pixel level clusters, as you can see here. And you can see that, for example, this purple cluster corresponds to the glomerulus, while perhaps this yellow cluster corresponds to here this purple and green distal tubule. So you can recapitulate some of those findings at the cell level as well. Then to add another level of different level of analysis beyond the pixel level and the cell level, we also integrated some spatial statistics. And I was lucky enough to work with some of the amazing people from the Robinson lab in Switzerland on a guide of how you can use spatial statistics that have mostly been used in geospatial sciences so far to actually get something out of spatialomics. And we have an online guide where you can see how this can give you an additional layer of information. So in spatiomic, we also have some functions implemented that allow you to do things like hotspot detection or neighborhood composition analysis to get more out of your tissue. Yeah, I think I'm running a bit short of time. So yeah, I had a couple of more slides how we scale beyond PathoPlex that I'll just super quickly go through. So I'm also part of the Spatial Proteomics Consortium within the SC-World, which is kind of the Python equivalent of Bioconductor, just to see how we can expand all of this further and also coalesce on kind of the same data formats and some of best practices. And then I had a couple of slides on how a recent development that we had on generative AI can actually help you with more parts of your experimental workflow that are not covered by imaging technologies. But I think there's no time for that. So yeah, I'll just point you to this publication, this correspondence of ours that came out last week in Nature Biotechnology. That basically proposes a framework for agentic AI to help with some more tasks of the experimental cycle. And with this, I would like to thank my lab, the Complex Tissue Analysis Lab, led by Victor Kuehl, and you can check out more of our lab at complextissue.com. And of course, all our funders, the PathoPlex team members, and all our collaborators. And last of all, thanks to all of you for taking some time out of your day to listen to me today. And I'll be happy to answer any questions you might have now.
Fantastic. Thank you very much, Malte, for a really, really interesting talk there. Excuse me. So with that, yeah, let's open the floor to questions. Does anyone have any questions they'd like to ask, either online or in person?
Yes, I'd like to ask a few questions. Firstly, have you tried any supervised method as well?
Any supervised methods?
Yeah.
Yeah, I mean, we sometimes combine already some pre-segmentation with this. So for example, sometimes we might just be interested in what is happening in the proximal tubule, and then we can use some staining that is specific to it to create a compartment mask where we just get that part of the kidney out, and then we can perform kind of our unsupervised analysis within that part. So I think we've tried it at that level to kind of pre-segment some parts and then combine information from different levels.
Also in like your cell masks, I saw that you have created an extended cell mask as well, like apart from the nuclei mask and the dilation radius of 10. So I wanted to ask, like, how do you adjust that?
Yeah, I think this currently has to be manually checked based on the tissue you have. We don't yet have any automated way to do this, but of course, this is an interesting research direction. For our manuscript, we mostly focused on the pixel level analysis, so we are still kind of expanding a bit in kind of the cell level and spatial statistics direction. And I'll definitely have a look if there's something out there, or if you can point to it to help kind of determine those parameters.
Thank you. No, I'm still working on it, so I'll share like if I get any. Thank you.
Thank you.
Brilliant, thank you. And can you also just remind us, sorry, Malte, so in terms of the subcellular resolution you get in, what is the resolution? What the highest you've got there?
So if you use kind of the latest generation confocal microscopes from Zeiss, you get a pixel resolution of about 80 nanometers, which corresponds to an optical resolution of about 110 nanometers.
Okay. No, brilliant. So and so in terms of the images that you showed in the, I think it was 90 different runs. How long does it take to the actual imaging side?
Yeah, so it depends a bit on how you do it in your lab. Theoretically, you can do about one cycle every 6 to 8 hours. So depending on your setup, you can do up to 3 in 6-7 hours. So if you work with 2 people in a shift, you can scale this up much further. But it also depends, for example, on which species are you using for your antibodies, because I think like we made a lot of progress in the first 30 days because we could combine different species and not have cross-reactivity. So we probably got the first 50 or more antibodies in less than a month, just with one person working on this during normal working hours, and also fulfilling some other duties during that time. But then, because we wanted to really scale this later, we ran out of multi-species antibodies, so we had to use always some rabbit, and then we had to go to one marker per cycle, and it took a bit longer. But I think if you plan this and optimize this, you can get about 20 markers in a single work week, standard work week for a single person. So if you do 2 weeks that way and have a plan, that you can get to 40, which I think, if you think about how long it would take else with other technologies to go through each sample, because remember, if you do this with this technology, you can do this for 80 samples at a time, 40 to 80 samples at a time. So even with some others that do single step staining, if you can only do one sample at a time, it might even take the same time or longer. So we are trying to work on further streamlining, and ideally, of course, you would sometime combine liquid handling with automated imaging. But already, I think this is within, kind of if you think about how this might look like in clinical translation later, you might have a tumor board that meets once a week. So if within a week, with people working shifts in pathology, in a pathology department, you can get 30, 40 proteins, I think that can already be quite useful.
No, brilliant. No, thank you. So, Chachi, I think, yeah, of course. Feel free to switch your camera.
Thank you for the presentation. My question is, what was the motivation for moving from the traditional segmentation-based methods to your pixel-level clustering? So did it come from some limitations you encountered in segmentation, or is it it's a new conceptual goal of them? I think you were trying to discover the gradients between these molecular interactions.
Yeah, thank you for the question. I think a lot of the motivation for us comes from the signaling side, and especially phosphorylated targets, because from the biological questions that we deal with, since we're very interested in like immunological damage and those patterns that drive kidney diseases, if you have a phosphorylated target, two things are important. First, that you can actually identify your signal at a cell level. And if you have a phosphorylated target that is not super intense and might only be present in a couple of pixels, if you average your signal at a cell level, you might not be able to detect any positivity at all, or might not really get a very wide dynamic range of how active perhaps a pathway is. And the second is that also where this is expressed in the subcellular, at the subcellular level, and in which compartment it is located is quite important. So I think those things served as kind of the main motivation to try it out. And of course, then this allows you to also identify more co-expression, where you could identify organelle crosstalk, or perhaps if in the nucleus you have condensation of the DNA or not, and similar things. But I think kind of the main reason was that we wanted to identify signaling in a better way.
Brilliant, thank you. So do we have any other questions from anyone in person? No, good. Okay then, well, I don't think there's any more questions from anyone in person. So if there's no one online who's got any further questions, we'll bring the seminar to a close then. But Malte, thank you very much for a really, really interesting seminar there. It's I've definitely learned a lot over the last 55 minutes. So thank you for that.
Thank you very much for organizing, for providing the opportunity. And if any of you still have a question that you might only think of today, later when having dinner, feel free to email me or email my PI anytime, and we'll be happy to engage.
Fantastic. Thank you. Thank you very much. And just before we close, just a quick reminder to everyone. So we actually do have we have another seminar actually scheduled for next week. So Monday, the 17th of November. It's going to be a talk by Professor Jana Lipkova from UC Irvine in the US, and her talk is titled "Too Many Models, Too Few Benchmarks: and addressing the benchmarking crisis in AI pathology. So I hope to see you all there. But thanks again, Malte. That was a really, really interesting seminar.
Thank you.
Thanks, everyone.